I'm just delighted to be here today. My goal as governor of New York ACP was to restart and boost up our district meetings because they were starting to fall by the wayside. And it's really important to be doing these educational things on a local level and be able to network with each other and become one community. So it was really exciting when Dr. Falsey agreed to do the talk today on such a the most timely topic of our lives right now in medicine, on the COVID vaccine. For those of you who don't know Dr. Falsey, she and I did our residencies together at the University of Rochester, so I've known her and admired her for many, many years. She went to medical school at Vanderbilt, and she did her residency here at the University of Rochester, and then her fellowship both at the U of R and at Yale in infectious diseases. She is a maven from the beginning of her research on respiratory syncytial virus and has expanded to doing research on other viruses such as influenza, parainfluenza, coronavirus, obviously, and human metapneumovirus. And she has done a bunch of vaccine studies over many years and has brought vaccine respect and to our entire Rochester community. And for that, we've been extremely grateful. So without using any more of her time, I want to very much welcome Ann Falsey to our session tonight. Ann, you have to unmute yourself.
Okay, now I gotta get back to my slides. All right, so I'd like to thank you for that kind introduction. It's, you know, I like to say it's either a really good time to be a respiratory virologist or a really bad time. So I'm not sure where I sit. You ask me every other day, I'll change my mind. So, you know, we are going to talk about COVID vaccines, how we got to where we are right now and where we may be going. These are my disclosures.
Dr. Falsey, just quickly, if you could tap share on your screen. I'm not sure if it actually, I don't see it on mine, so sorry to interrupt. Just want to make sure.
Oh, I'm glad you did. Technical.
Perfect. I see your, I see the slides. The slideshow just hasn't started yet.
Yeah, it's weirdly not letting me do my. Okay.
Oh, now I see it.
Okay. Oh, and now I see everybody else at all. Okay. So I don't probably have to tell anybody how bad this pandemic is, but I thought that this is useful just to look at, you know, as a respiratory virologist, every year we look at the MMWR P&I report, and every year you see this sort of sine wave of the usual respiratory crud that happens every winter. And when you get a spike that exceeds the expected mortality, that is usually associated with an influenza epidemic, not a pandemic. And in 2018, we had a pretty robust flu year. And then you look over here at 2020, and it just absolutely dwarfs anything we've ever seen with the flu. So it just puts it in perspective, as if we really needed to be reminded how bad it is. So it's hard to talk about vaccines without first talking about the virus. And I think you probably are all very familiar with this cartoon of these little knobs sticking out. So it is an enveloped virus. It's single-stranded RNA, and the spike protein features very prominently. It is important functionally, and it's also important immunologically. And this is a close-up of the spike protein. It is a class 1 fusion protein, just like we find on my first love, RSV. And it binds to the ACE2 receptor on the human epithelial cell. And shown here in green of this electron micrograph is the receptor binding domain. And it acts like a little finger that kind of sticks out. So this is a cartoon, and it shows the pre-fusion spike protein. These are the ACE2 receptors, and this is the little thumb. And as it sticks out, it joins with the ACE2 receptor. And when it finally fuses, the fusion protein undergoes a significant conformational change, and then it brings the two cell surfaces together. And you can see that if you had an antibody that blocked the attachment of the ACE to the spike protein, it would inactivate the virus. Normal vaccine development requires 10 to 15 years. And just shown here are the various stages of vaccine development, all the way from when you're in the test tube to a phase 3 clinical trial and everything in between. Phase 1 is generally when you're testing very small numbers of very healthy people. You start young, you move up the age groups, and that's where you select your vaccine construct and also the dose. And then in phase 2, you go from a couple of hundred patients to a couple of thousand. And then finally in phase 3, after you've selected the proper dose, the best construct, you put it into many people to see whether that immune profile actually translates into it being an effective vaccine that prevents the disease. Now, normally these are done sequentially. What has happened, and we try to reassure the public that even though things have moved very rapidly, that none of these stages have been skipped. They have been accelerated, they are overlapping, but they have not been skipped. And in particular, the manufacturing is being done at risk. So normally a company wouldn't manufacture millions of doses of a vaccine before they knew that it works. But here the government has taken the responsibility, and although I hate the name Operation Warp Speed, it has actually been useful to get this program moving rapidly. So these are the 3 types of vaccines that are currently in phase 1, 2, and 3. The messenger RNA vaccines are first out of the gate. And that's where I should say that all the vaccines are aimed at making the body have an immune response to that spike protein. So the RNA vaccines deliver the spike gene itself into the body so that the body's own cells then make the protein. The vectored vaccines, AstraZeneca and Janssen, they use a non-virulent virus, kind of the Trojan horse approach, to bring in that same genetic code, and then your body's cells also make the protein on the surface. And then the last approach is where the spike protein is made in the laboratory. And that's sort of the traditional vaccine approach, and that's what Novavax and Sanofi Pasteur are doing. So again, just the messenger RNA, you take the gene that codes for the desired antigen, in this case, the spike protein. That's what's injected. The RNA gains entrance into the cell. The message is translated into protein. The antigen is displayed on the surface of the protein and interacts with your immune system. The advantage here is that the protein, because it's made by your body, is displayed in a natural conformation. And so that you don't get any skewing of the immune response because you're displaying a lot of what we call non-neutralizing epitopes. So these are some data from the Moderna phase 1 trial, and it shows what we call the reactogenicity here. And we have vaccine 1 and vaccine 2. And here are the systemic side effects, and here are the local ones. And it's broken down by the doses. They tested 25, 100, and 250 micrograms. So severe reactions are shown here in yellow, and I think you can tell that most of the reactions were occurring after the second vaccination. And I think, you know, if anybody has had your RNA vaccine, this is what people are experiencing, is that the second vaccination tends to be a little worse. And if you look at the different doses, it's really with the 250 microgram dose that they were seeing more in the way of severe reactions. This is the typical immunogenicity data, and shown here are the various neutralizing titers, and then it's broken down by the different dose groups, and these are days after vaccination. And what you can see is after one dose, you got some antibody production, but it was really after the second dose, that booster, that you got this nice rise in neutralizing antibody. And you can see that the 100 microgram dose was maybe a little better than the 25, but there really wasn't too much difference between 250 micrograms and 100. And so because it was significantly less reactogenic for the phase 3 trials, and now what is available EUA is the 100 microgram dose. For Pfizer, they evaluated two different constructs. They looked at just making the receptor binding domain itself. And then they also looked at providing the gene for the whole protein. Because encoding it in lipid actually helps it get into the cell better, these vaccines, both Moderna and Pfizer, are coated into lipid nanoparticles. And this just shows the data. We were very fortunate here in Rochester to participate in the phase 1 study. And what it shows is this is the B1 construct, that's just the receptor binding domain. And this is B2, that's the whole gene. And interestingly, it was significantly less reactogenic. Yeah, I would have just guessed that it would be the other way around. And the 30 microgram dose was chosen. I won't show you the immune response, but it gave the best immune response, and the B2 construct was chosen because it had a better side effect profile. Oh, here's the immune. I'm going to skip this. Okay, so the phase 3 trials began at the end of July. There were 99 Moderna sites, 149 Pfizer sites. There were 3 international sites. Moderna had 30,000 people, Pfizer had 45,000 people, and they were randomized 1-to-1 to vaccine versus placebo. The endpoints, the primary endpoint is prevention of any symptomatic COVID illness that's proven, PCR proven. And the time to analysis was really event driven. So the sample size was constructed with a certain assumption of a certain attack rate to achieve 150 COVID illnesses. So you could then look at how many occurred in the vaccine group, how many occurred in the placebo group. And they initially estimated it would be 6 months. It was really just under 4 months because the country began to be on fire. Just a word about AstraZeneca. We are currently just finishing up that study here in Rochester. So the AstraZeneca vaccine was developed in Oxford, England. It is a replication deficient chimpanzee adenovirus that expresses the spike protein. And it's replication deficient in that it's a live virus, but it can only replicate once. So you cannot possibly get a chimpanzee infection. And in addition, a chimp adenovirus is not pathogenic for humans. There actually was a fair amount of experience with this vector. And so when the pandemic began, they just swapped it out. And immediately in April of last year began their phase 1 study. They didn't actually use saline, they used Menactra as their control. And they initially planned for this vaccine to be a 1-dose vaccine. They did do some boosting, and kind of after they began their Oxford trial, they realized that they were going to have to do 2 doses, just like the RNA vaccines. And so sort of midway through, they started doing the boosting studies. So the current phase 3 study, which is really just wrapping up, people over the age of 18 was anticipated to be 30,000. So 2 doses, as I mentioned, of 5 times 10 to the 10th particles, given 1 month apart. Different than Pfizer and Moderna, 2/3 of the vaccine study population gets the vaccine, 1/3 gets placebo. Very similar endpoint, the prevention of symptomatic COVID. So the US study opened at the end of August. It will be fully enrolled this week at 32,000. And hopefully we'll have an interim analysis in a couple of weeks. So lastly, the protein-based vaccines I mentioned, traditional vaccine method, it's wave flu vaccine, hepatitis B. You basically make the protein in the test tube and purify it. But you have to actually be quite careful that in doing that, you do not disrupt the conformation or the important immune spots, what we call epitopes. And frequently, adjuvants are needed to sort of goose the immune system to make a good antibody response. RNA is naturally an adjuvant. So what's moving forward are Sanofi and Novavax. Sanofi is using 2 different adjuvants. It's very analogous to flu block. The phase 3 trial was anticipated to be started back in December, but it was put on hold because their phase 1 data did not look immunogenic enough. So they've gone a little bit back to the drawing board to really make a better immune response. Novavax is a nanoparticle. They have a novel adjuvant called Matrix M. It's also an oil-based adjuvant. It's very similar to the licensed Ebola vaccine that they have. And that phase 3 trial began right before the holidays. And it is placebo controlled, but it's getting harder and harder to do a placebo controlled trial. So hopefully they'll get it fully enrolled. So final analysis for Pfizer, they had 43,000 people enrolled. They ended up with 170 cases of COVID for their final analysis. The endpoint was determined for any case that occurred at least 1 week after the second vaccination. And amazingly, it was 95% effective to prevent symptomatic COVID infection. And good efficacy, 94% even in older volunteers, and was quite effective to prevent severe cases. And so as we know, they've submitted to the FDA and received their emergency use authorization. And it seemed almost too good to be true until Moderna came along and really validated those findings. 30,000 people, their endpoint was a little bit different. It was 2 weeks after the second vaccination. And they found 94% effective to prevent symptomatic infection. And it was 100% effective to prevent severe disease. And it also has passed through and achieved emergency use authorization. And although these things are, initially it was like, wow, this is just too good to be true. We were very thrilled. We worked on Pfizer. It was, very hopeful that, gosh, now we have a vaccine. Like I said, Moderna validated it, but these vaccines are working in a way that we don't completely understand. They're achieving protection so quickly, it can't be based on the usual neutralizing antibody. And as I mentioned, RNA is a potent adjuvant. It stimulates the innate immune response quite vigorously. I think that may be why people get some pretty robust reactions. So we have much to learn about how these RNA vaccines are working. And, it may be that in the future, all vaccines are RNA vaccines. The interim analysis, some of you may have heard, came out with the Oxford vaccine, which is basically the AstraZeneca vaccine. It was 70% effective to prevent symptomatic infection and was 100% effective to prevent any kind of severe disease. Nobody was hospitalized and nobody died in the vaccine group. And clearly they had severe cases in the placebos and even one death. You may have read about this high dose, low dose stuff. I'm not going to go into it, but if somebody has a question, it basically, there were so many variables in that group. It probably was not the dose if it was real at all. It was a very small subgroup analysis and really probably should not have been reported. It's just too interim. They have partnered with Serum Institute of India and are making billions of doses. Oxford has pledged a no profit and they have an EUA now in the UK. And the advantage of the Oxford vaccine is it's going to be quite a bit cheaper and it doesn't have the freezer requirements, particularly Pfizer with the -80 freezer issues, but even Moderna. And so I think that people are very hopeful that this will be a good vaccine for the world. I don't have any slides on the Janssen vaccine, but it's also an adenovirus vectored vaccine. That trial has fully enrolled. They are anticipating a analysis in the next couple of weeks. And so hopefully, within a month or two that the Janssen and AstraZeneca vaccines can supplement the current vaccines. Just a little bit about the process, FDA emergency use authorization. It's actually kind of rare for vaccines. They did it in 2004 for anthrax. And as you can imagine, it's a faster review that's less stringent and does not have to meet all the requirements for full licensure. You may have heard about the VRPAC meetings, and this is Vaccine-Related Biologic Products Advisory Committee. And these are experts that are completely independent. They offer advice, and it's actually a public hearing that you can listen to if you want, where they consider and they debate, they offer advice to the FDA, but ultimately the FDA makes the authorization. So we're now in the phase of vaccine distribution because we do have the RNA vaccines. And this is just a broad schema of where we are when doses are quite limited, and that's where we are now. And we have these issues of cold chain that you really have to be very targeted and strategic on how you roll it out. Once there's large numbers of doses available, then it becomes logistics on how to really coordinate making vaccine distribution centers. And then finally, the last phase is when there's continued vaccination, but the supply is really sufficient to meet demand and you have the infrastructure to deliver. So I think we're all aware that we're currently moving through these very first phases, phase 1A, healthcare workers and long-term care residents. We've now begun phase 1B, which is essential workers, including law enforcement, some teachers, other essential personnel, and people over the age of 75. Now, here in New York, we've moved on to 1C very rapidly. I don't think that we have the infrastructure and the supply to actually deal with that. We have a lot of people calling, wanting their vaccine, wanting to make appointments, and it's still in quite limited supply. Just to sort of wrap things up, because I want to leave plenty of time for questions. I'm sure there are some interest in side effects. And overall, these vaccines are turning out to be very, very safe. The most common side effects are consistent with what was observed in the clinical trials. They do have some reactogenicity, so it's important to plan that on your first or second, particularly your second vaccination, you may have fevers and chills and may actually need to not go to work. Interestingly, 52% I should say that there's this network, the Vaccine Adverse Event Reporting System called VAERS, a little over 6,400 have been reported, and 52% have been in white non-Hispanic individuals. And it's a funny way of saying it, but that means that 48% are in diverse populations, and we know that it seems skewed, but there's more to come on that. And then there's certainly enhanced surveillance for anaphylaxis. It gets into the news a lot, but it really is not that common. 36 confirmed cases. So the crude estimate is 6.1 per million doses administered. Interesting, I was just on a phone call listening to this stuff about anaphylaxis, and I didn't realize anaphylaxis is more common in women. I don't know why, and this is holding true. Only 2 cases have occurred in men, 34 in women. The median time to onset is 10 minutes. 89% occur within 30 minutes. 83% of the individuals had a history of allergies, and 50% of these individuals actually had a history of anaphylaxis. The other thing that a lot of people are asking about is what is the effect of these new variants on the vaccines? And with any RNA vaccine or RNA virus, they mutate at a relatively high rate. Many of the mutations are silent. Some do translate into amino acid substitutions, and then whether or not it's important for the virus or not deserves further study. And this is showing when the virus first emerged from Asia. The strains are indicated here in blue. When it got to Europe, there was a mutation. And interestingly, the map over here shows that that strain or variant that ended up in Europe really has become the dominant pathogen worldwide. And so there must be something about that virus that makes it more fit or more transmissible. It's not more virulent, and they actually have done studies in animals and have showed that this variant, which had a mutation at amino acid site 614 in the spike protein, actually did allow the virus to replicate more efficiently in the upper airways. And so that makes sense that it would be just more transmissible if you've got a lot more virus in your nose. So now we're hearing a lot about the variants coming out of the UK and also South Africa. And so shown here on this slide, these are the divergence from the root, the sort of ancestor. And then over time, you would expect that there would be mutations and variations. And this is now this new lineage that's being reported out of the UK, the B117. And you can see that, you know, shown here, it's really quite far apart from the other lineage. And it's replacing the, it's spreading at a rapid rate. And so is it simply that the United Kingdom's on fire and everything is spreading rapidly? But it does seem to be taking over as the dominant strain, suggesting that it's got some advantage. And it isn't more virulent that anybody knows. And these are the amino acid substitutions. So there's 20 sequence changes that actually led to 14 amino acid changes. And shown here in blue is the amino acid. This is the N501Y mutation. And that one has also been identified in the South African strain that seems to be becoming the dominant strain there as well. We don't have complete explanations, but this again is the spike protein. And right here is this mutation at 501. And it seems to be where in an important area on the receptor binding domain. And there is some evidence, it's very early evidence, that perhaps it increases the avidity for the attachment of the virus to the ACE2 receptor. There is, I don't have a slide for it, but you know, everybody's question is, well, you know, are the vaccines going to work against this new strain? And one of the advantages to a vaccine that encodes for the entire spike protein is that there are multiple epitopes on that protein. And so your response to that immunization is not one single type of antibody. It's polyclonal. So you are making antibody to lots of different places on the spike protein. Now, you know, making antibody to the receptor binding domain is probably important. It's an important functional attribute. And there was just some very early data released from BioNTech Pfizer, where they did take serum from people who had received the vaccine, and it appeared to neutralize this variant similar to the original strain. So that's good news. But I do think we have to be prepared that there will be some evolution of the virus over time. There may be adjustments that need to be made. And one of the nice things about this new technology, although, you know, some people find it a little scary, is that you can rapidly change what you're doing. It's very amenable to, you know, just putting in a new mutation so that your vaccine mimics the circulating strains. So in summary, just to reassure people that the vaccine's been developed in record time, but no steps have been skipped. They are very safe, but caution should be used in patients who have a history of anaphylaxis. And we're not quite sure what is the allergen, whether it's polyethylene glycol or something else in the vaccines, but they should be used very cautiously in somebody with an anaphylactic history. It is expected that the vaccines will retain efficacy to these new strains. Durability is completely unknown. We don't know whether we'll need booster shots. Currently, vaccine mixing types is not recommended. There's no safety data. The pediatric studies are coming. Hopefully we will be doing some of those here at the University of Rochester. Pfizer did include adolescents in their approval, so that is approved for people over the age of 16, greater than or equal to 16. Moderna is currently at 18. We're hoping the pediatric study for Moderna will start in March. There's really minimal data in pregnant and breastfeeding women. There were, of course, in any big vaccine trial, even though you're not supposed to get pregnant, there were women that got pregnant, and there didn't seem to be any ill effects. And the American College of Obstetrics and Gynecology, their approach is that the vaccine should not be withheld for women who are meeting CDC criteria. And all of life is risk benefit. And, you know, if you have a pregnant woman who is a first responder, particularly somebody who might have some health condition, the risk of COVID may certainly outweigh any theoretical risks of in pregnancy. So I'll stop there and see if anybody has any questions.
Thank you so much, Dr. Falsey. This is Amanda, everyone. I am going to read some questions. We had some come in, and we also had some submitted upon registration. If anybody would like to ask a question verbally, please click on the participants button at the bottom of your screen. And there is a raise hand button, a little blue raise hand button on the bottom of that participant list. So I'm going to start with Dr. Polsino was wondering if you could comment on why for this virus an mRNA vaccine was identified as an approach, but not for other viruses in the past.
So it has been in development, and there was a flu vaccine that's an RNA vaccine. But you know, it was sort of moving at the turtle pace that I showed you, the 10 to 15 years. And it is a technology along with the vectors that was, there were other vaccines in the pipeline using RNA. And so they just swapped it out and said, all right, we're ready to go. In particular, Moderna had a Zika virus vaccine in development with the RNA vaccine approach.
Thank you. Dr. Cleary was, well, first of all, she thanks you for an excellent presentation as well. But she was wondering, do you think that the stringent requirements for maintaining the sub-zero temperature for Pfizer vaccine will be sustainable in the field? And if not, will it impact the efficacy when it is in widespread use?
Yeah, it's a good question. You know, they will be doing some further testing on Pfizer as to whether or not that is necessary. And, you know, the long-term storage needs to be at -80, but once you deploy it to the field, does it really need to be that? Everything moved so fast that that's where they kept it. So that's what's approved. And it may change in the future when they do further stability testing. But, you know, it's still not going to be the greatest vaccine for the developing world. You know, I think in this country, most places can handle it. I think it will create problems if they, you know, as far as sort of having it available at your local CVS. It's just got issues. Moderna, regular -20 freezers. For the world, neither one of these vaccines are great. And that's why a lot of people are pinning their hopes on the vector vaccines that just simply have to be refrigerated, or the protein vaccines.
Understood. Speaking of that, is there a particular reason when somebody was wondering if there's a particular reason why the mRNA vaccines have to be kept at such a low temperature?
Yeah, probably. You know, okay. They're not stable at, you know, higher temperatures. But again, I don't think they did sufficient testing to know how unstable they are for how long. They just kept them at the absolute coldest so they would be sure of their potency, and they moved them along. And so those stability and stringent requirements, I don't see a reason that the Pfizer vaccine should really be different than the Moderna vaccine. Honestly, you know, they have slightly different lipid coatings. And, you know, things that are proprietary, but, you know, they're almost the same vaccine. So I don't know that Pfizer did their stability testing, and hopefully they will soon.
Okay. I'm going to kind of combine a couple of questions here. A bunch of them came in about really who should get the vaccine. You know, they're getting calls from patients asking, or they're wondering themselves for recommendations for patients. So you already addressed pregnancy. You know, they're getting calls regarding use of steroid inhalers, immunocompromised individuals. I think one came in here, those vulnerable to autoimmune system syndromes and diagnoses. You know, there's a lot going on whether it could trigger responses or patients are feeling anxious about it. So what are your thoughts and recommendations for that?
Yeah, I mean, there's two different ways to be at risk for COVID. One is you're at risk to acquire it because of your job. You're just out there. And, you know, as much as we think that, you know, young, healthy people don't get that ill, they get pretty sick, some of them. It's sort of, you know, it's a real crapshoot. Some people are asymptomatic and some people are quite ill. And so I think that the vaccine really should be going to the teachers, the first responders, people that by their circumstance have essential jobs. Then when you get to regular people who it's just their increased risk of developing more severe COVID, we have, you know, the very elderly, you know, that's the over 75-year-old group. And then people over 65 and people with chronic conditions. That's a lot of people. And, you know, I can appreciate that you may have a difficulty triaging that. And my own personal feeling is that people who are, you know, this virus acts differently. You know, I've been a respiratory virus my whole life, a virologist my whole life. And, you know, RSV and flu, it's COPD, it's heart failure, it's that, you know, it's that kind of thing. Whereas not so much here. Here it's hypertension, obesity, diabetes. I think that the obese diabetic is really at very high risk. So, you know, I think though that practically with deployment, it's going to be awfully hard to manage that. You know, primary care providers, you know, you can recommend that the patients you're most concerned about get vaccine, get in line, advocate for them. But I don't know that you're going to have that much control over who gets their appointment when. So, you know, I certainly would hound the people you feel to be at highest risk to not have hesitancy and to go get the vaccine. But then they're going to have to, you know, it's very hard with these clinics and there's just not enough vaccine. Regarding autoimmune disease, it's always been a concern that somehow having an autoimmune disease may be kicked off by getting a vaccine. People feel that way sometimes about flu vaccine, about the shingles vaccine. I think the risk is very low that there would be any harm. And I think if you're somebody with an autoimmune disease, you may be at increased risk for severe COVID. So I think the risk of getting COVID outweighs the risk of any detrimental effect from the vaccine. Regarding immunocompromised hosts, they were all excluded from the trials. And I think we can infer from all the other vaccines that they will be less effective in that population, but I wouldn't withhold the vaccine. I think if they are certainly at risk. So I think they should be vaccinated in the group of people who have underlying conditions.
Regarding another group, particularly the older population, you know, were there any that were included in these studies that you know of? Any of these studies that had dementia, MS, or, you know, were maybe over a certain age, like 75, over 75 or over 85? Were there a number of them involved in the studies? Do you know?
Yeah, so not as many as you might like. We probably do need specific studies in those populations. You can imagine doing informed consent when somebody cannot consent for themselves is problematic in a phase 3 trial. So I don't think there were many people who could not sign their own consent, but there were some nursing home patients where families consented for them, but it's just so logistically hard to do that wasn't done too much. But older people, all the trials were required to have a significant elderly population in the trial. I forgot what the last part of your question was.
Oh, it was just basically the either age groups or by diagnosis like dementia or MS. So I think you answered it.
Well, demyelinating diseases are always they are excluded, particularly with the AstraZeneca trial. They were on pause for a neurological event. It was labeled as transverse myelitis. It turned out there were 3 events in the UK study, and they were all labeled transverse myelitis. And one was in somebody who had pre-existing multiple sclerosis and didn't actually know it. And so that was not related. Weirdly, one was in the Menactra arm, so that clearly wasn't related, but you wonder what's going around the UK giving people transverse myelitis. And the third case that actually led to the study pause, having seen the really granular data, I'm not sure what that person had. Fortunately, it sort of resolved in 48 hours. It was something. The MRI wasn't quite normal. They had tripped while jogging and sort of wrenched themselves. I mean, we really don't know what that was. But right. Specifically, most vaccine manufacturers will never allow anybody with a demyelinating disease or Guillain-Barré into a trial because it could grind it to a halt if something happens. And I think that would be a discussion that you'd have to have one-on-one with individuals about risk benefit. It would make you nervous to vaccinate somebody who has had a history of Guillain-Barré with really anything. It's an immune stimulated disease. So I don't know what there is.
Yeah, that actually brings up, I have another question regarding, when to get different vaccinations, but one came in, or what, no, let me move on. But one came in actually about Guillain-Barré and mentioned that the 1976 swine flu vaccine was associated with it occurring up to 6 months after. And they're wondering what, if anything, is known about the possibility of serious late reactions, like 3 to 6 months down the road. We
don't know anything because we don't have any long-term, and that's why when you are telling people to get vaccinated, the clinics, and they're giving out the cards that have the number for the VAERS reporting, and post-marketing reporting is very important because rare events cannot be known in a trial of 30 or 40,000. You can only know that it didn't occur in 45,000 people. You go to millions of people, it's different. With the swine flu and the increased rate of Guillain-Barré, it's always important to remember, the difference between sort of absolute and relative risk. While the relative risk may have doubled, the absolute risk is still like 1 in a million. Now it's 2 in a million. They're still very uncommon events, and COVID's very bad and very prevalent.
Yeah, thank you. What I was trying to get to before and I scratched was there's been a couple questions about actually getting the vaccine. So the first question is, if somebody has COVID already and they're eligible, should they be getting it, or how long should they wait before getting, after they test negative to be getting that?
Yeah, and that's a really good question, and we really don't know the answer to it. I do think that there is evidence of some immunity from natural infection, and how long it is, we don't know, but the recommendation currently is to wait at least 90 days after your infection, and that is in part because vaccine is in very short supply. And so, that vaccine might be best used in somebody who is completely naive and at risk. And so, I can't say that it's absolutely contraindicated, but it's not contraindicated, but, I just would recommend that somebody forego the dose because they're relatively protected for some time.
Okay. What about in between the first and second vaccine? So somebody or dose, somebody already has received the first one and either they get COVID or perhaps have a reaction like, a rash requiring prednisone.
Or they
Should they get
just go to Florida and they miss their appointment. And so, the dosing intervals of almost every vaccine are quite empiric. And, why 3 weeks for Pfizer, 4 weeks for Moderna? That was not based on anything. Pfizer was in a bigger hurry. So we do know that the longer the interval, probably the better the immune response. That for whatever reason, your B cells take a while to mature and develop that optimal memory response. And so I mentioned the sort of the high dose, low dose stuff that went on in the, what really happened is a lot of people that got the half dose by accident, I will inform you, the interval was longer. And so probably the real reason they had a better immune response is that there was a longer interval. So immunologists know that, but when you are in the middle of a raging pandemic that's killing people, you don't want to wait 6 months for the ideal immune response. You want to boost and get a better immune response more quickly. And so, waiting 4 to 6 weeks is probably the ideal time to come in with that second shot. We are recommending that we stay studied. So Pfizer, it's 3 weeks. Moderna, it's 4 weeks. But if somebody was delayed a week or two, it should not affect the immune response or their protection at all. If anything, it might even be better.
So even if it was a delay of more than that,
Yeah. Mm-hmm.
Okay.
You wouldn't give them an extra dose. And in fact, you really don't want to give them extra doses. There have been a few people that were in the Pfizer trial and then decided they'd go get the EUA vaccine as well. And so they ended up with multiple doses and had pretty severe reactions. So I think the lesson there is we just need to stay with the evidence. The current evidence is that there's 3-week interval for Pfizer, 4-week for Moderna, but I would not be dogmatic about that. And you can reassure people if they're late with their second vaccinations, or particularly the way the Biden plan is being proposed, which is, well, we're going to vaccinate everybody, and then we're going to just worry about the second doses later. They feel like that manufacturing is going well enough that they will be able to get those second doses at the proper time. But let's say they don't, there is a delay. I really don't have any concerns that it would be less effective.
So particularly, I have a question that a couple of people asked. I think it was involving what is the recommendation for patients who received the first mRNA vaccine but who developed urticarial rash requiring prednisone? Should they proceed with the second vaccine?
Yeah, it's a good question. I think in my mind, it depends on, the immediacy of the reaction. And you can work with an immunologist on this. I mean, allergist. In that, I've seen people who get sort of itchy, bumpy rashes, a day or two later. And I would be less concerned that if somebody developed immediate hives, like within that first 10 to 30 minutes, that's an immediate IgE mediated thing. And I would be reluctant to give that second dose. If I did do it, I would do it in a healthcare setting, in a hospital,
Right.
Okay. I would worry. But
And
not with just a run of the mill, rash the next day.
Understood. I'm not sure if you know this, but do you happen to know what percentage of the vaccines already administered have been Pfizer versus Moderna?
I don't.
Okay.
I mean, I would think that, a little more Pfizer because they got their EUA a couple of weeks earlier.
Gotcha. And I know you've mentioned, or not you mentioned, you presented to us how the virus has evolved and changed with these new variants. How do you think it will evolve over time? Do you agree with the recent Science paper, if you're familiar with it?
Well, I'm so sorry I haven't read the recent science paper.
Okay.
Busy unblinding all our study volunteers who are getting EUA vaccine. I think what I could say with certainty is it will continue to evolve. So the coronaviruses mutate at a slower rate than flu, and whether or not it's going to hit antigenically important spots, we don't know. I think that, I can't imagine that we won't have partial immunity, even if it does mutate. And, the seasonal coronaviruses have been around for quite some time. And, you get a bronchitis, you get a cold. There are things you get in childhood. There is no 100% permanent lifelong immunity to any of the respiratory viruses. It just doesn't happen. We get them over and over again, but you never get, you wouldn't expect to get this scary, severe infection. So whoever read the science paper, what did it say?
Well, I read the New York Times take on the science paper. I didn't read the paper itself, but it said that some of the viruses that we have now actually may have been what spurned pandemics in the past, but that as we get immune tolerance to them, they just become more common like other coronavirus colds. And that might be the way that this evolves over time once people either get the vaccine or the natural infection.
Yeah, I mean, there will be immune pressure that, when everybody's walking around with antibody, whatever virus manages to survive in your nose and spread to somebody else will likely be resistant to the antibodies that you have. But it may be that they're much milder. In addition, if you achieve herd immunity and a lot of people have immunity, it just like you said, that somebody will get a cold, but they won't get severely ill. So there's a lot yet to come because it's a new disease, but I think we can look at the seasonal coronaviruses and have some idea that that's what we would expect this to do.
Can I do a follow-up question then? So would you expect, given that scenario, that we will be doing boosters like we do with flu every year, or do you think that it might be such that that won't be necessary in the long term?
I'm hopeful that it won't be necessary in the long term, but I think it's too new. I don't know.
Thank you. I haven't seen any hands raised, so if anybody else did want to ask a verbal question, I'll open it up to one more since we were a little late starting, but I want to be cognizant of and respectful of everybody's time. So I'll just pause. There are definitely a lot of other questions. I'm sorry we couldn't get to all. I definitely have a huge sheet. So I tried to get them all together, I guess. First of all, thank you, Dr. Falsey. Do you have any words for your fellow physicians who are trying to ease the mind of their patients or what to share with their patients?
Yeah, my recommendation is that now is the time to double down on your masking and social distancing. That the vaccines will get available. You will get one. It may not come next week when you want it, but you will get one. And don't get frustrated. And I also think that people who've been vaccinated need to continue to wear masks and distance, because we don't know what this does for asymptomatic infection. And so they could still transmit. And until we achieve herd immunity in the population, there could be vulnerable people that could get quite sick. Now, a couple of people were actually raising their hand, but they look muted. I don't know.
I didn't see that. I'm sorry, I can't see everybody.
So it's Esther and Keith. They have questions.
Oh, sure. Dr. Tansman, go ahead.
So if you got your first shot and the second one is delayed, how protected are you? I mean, your immunity starts, but I know they didn't quite look at that.
Well, if you can look at the articles in the New England Journal, it's sort of amazing. The protection starts to become apparent around 10 days after the first dose. They've got these nice graphs that show the placebos. And it looks like it's around 10 to 15 days. How protected are you? I don't know, but it's definitely starting after the first dose.
So my question was a little bit more theoretical. So you talked about these mRNA vaccines being active against multiple epitopes. And I'm curious if you know anything, I know this is out of your field, the mRNA vaccines against HIV, have they been tested? And is that something that we would expect would be better than some of the other trials?
I don't know. I honestly, I made a decision early in my career that I was going respiratory, not HIV. So the molecular, HIV has been so difficult. And, the problem is it integrates into the host cell. And so I'm going to just say, I don't know. I think the RNA vaccines are new. They have a lot to offer. And so maybe.
Dr. Falsey, thank you so much. And thank you to all the attendees who asked questions and joined the conversation. I personally, I'm not a physician, but I learned a lot, and honestly learned a lot to take to my friends and family who have so many questions. So to lead them to you guys and help with that, hopefully. So I appreciate it. And thank you so much for your time and a great presentation and great information. And thank you to all of those who joined. We did record this, so I'm gonna try to get that recording edited and sent out for everybody. Thank you to the doctors, the district presidents, Dr. David Heisig from Syracuse, Dr. John Fadima from Buffalo, Dr. Tiffany Polsino from Rochester, and Dr. Pedro Gonzalez-Zayas from the Southern Tier, as well as our governor from Upstate, Dr. Al Gason, organizing this. We can't thank you enough, Dr. Falsey. It was a great presentation. Thank you for your time. And be safe, everyone. We're so glad you were able to come. It was great to see faces and learn together.
Thanks for having me.